This
Good morning,
conference will
everyone.
now be recorded.
Good morning, everyone. First, I'd like to thank Elisa for giving us the opportunity to present here genOway. And what I would love to do today is to basically give you an overview of what we do with a focus on a model that we have with which display both human lymphoid and myeloid compartment. Before getting into the detail of our technical platform, so who are we and what do we do? So genOway is a company which developed preclinical models. We're in a business now since 22 years, and we provide models to 17 of the top 20 pharma companies, but also to many well-known biotech and let's say mid-sized pharma companies. We started our activity by developing model on demand. That was our main business up to 2015. And in 2015, we decided to add another arm in our activity. So we are still making model on demand. We developed, majority of the model we developed are knock-in models, which are humanized for certain targets. And we decided to take advantage of what we have done in the past, because we were within the first company to develop immune checkpoint humanized model by knock-in. We decided to take advantage of what we learn around those targets to also provide off-the-shelf models. But we have different type of off-the-shelf models today. Models which are humanized for the whole immune system. I will detail this later in the presentation. Models which are mouse having a mouse immune system in which certain targets have been humanized. These mice here display a humanization strategy which is really target dependent in order to make sure that we're going to keep the functionality of the target while keeping also its interaction with partners functional. And I will say a few words about it. Model for T-cell engagers. And finally, model for PK/PD assessment in a context where both the FcRn and thermoglobuline are humanized. Let's first say a few words about this model, which I'm not going to detail, but just to let you know what we have in the pipe. So genOway has developed a catalog of approximately 30 different immune checkpoint humanized models. This model here have the advantage of having a fully functional interaction between immune cells, stroma cells, and tumor cells, since we are in a syngeneic system. The humanization strategy has to preserve the functionality, of course, of the target, and for that we do a lot of de-risking in vitro to ensure that the contract that we're going to select would keep the target functional. But we also pay attention to keep the proper regulation of the target, because we all know that immune checkpoints are highly regulated and their expression level will also depend on the immune response which is going to be developed. These models have been used for different purposes. For example, BMS has used our VISTA humanized model in order to establish that VISTA was a ligand for PSGL1 and that this ligand worked in acidic condition, and as such having an antibody which worked under acidic pH would enable the blocking of these targets. Model has been used also in order to define combination therapy. This is what Alligator Bioscience has done with our CTLA-4 OX40 double humanized model, being able to show that the antibody goes on target, the bispecific goes on target and specifically at the tumor site and not and modulates immune cells at the tumor site and not at the periphery. And finally, one of the latest publications of our model was this design of a PD-1 CTLA-4 bispecific antibody done by AstraZeneca, where they take advantage of the fact that both humanized targets were well controlled in terms of expression level in our model, in order to develop a bispecific that will be first bind to PD-1, which has a higher expression on tumor microenvironment compared to CTLA-4, and the binding to PD-1 result into a binding to CTLA-4 with a lesser affinity, which overcome the cytotoxicity, which is known to be CTLA-4 related. So we have as such a panel of models which are humanized for different immune checkpoints. I'll be more than happy to talk about it at a later stage if necessary. But today, for our discussion, I would love to focus on the following. These type of models are well known. There's many models around, but I really insist on the fact that the quality of the model is really dependent on how they have been designed and whether or not we have kept in mind the biology of the target when designing those models. So any knock-in model is not, knock-in models are not equivalent in between. It's really dependent on how they've been made. The main limitation of these models is the fact that they are really restricted to one target. And should you wish to either modulate a target which has no mouse autolog or to try different combination therapy, then it's becoming a problem to use knock-in model, because then you have to anticipate all those intercrossing in between different knock-in models, which is time consuming and which most of the time is not in line with your own objectives. So as such, we also decided to develop in our platform model which display a human immune system. So the model that we are using in-house is called the BRGSF. So what's BRGSF and how this model differ to pre-existing model which are available on the market today? The BRGSF model is as follows. First, that model has been developed on a biopsy background. Why do I emphasize the background? Because you know that all the existing models were developed onto a NOD background. NOD mice are deficient for C5a. As such, the complement cascade in the NSG or in the NOG mice is not functional. In our case, the C5a is still wild type, and as such, the complement cascade is functional, and model could be used to assess, for example, mechanisms such as to assess therapeutics which work throughout mechanisms such as CDC and other mechanisms involving the complement system. The mice are on a RAG2 knockout background, which means they have no T cell, no B cell. They have no NK cell because of the knockout of the IL-2 receptor gamma common chain, and they do express sub-alpha from NOD origin, which provide a proper interaction with human CD47, and as such activate the don't eat me signal. So that prevents the phagocytosis of the human cells by the mouse phagocytes. Finally, we've added into this model a mutation for a gene called FRIC2. That mutation results in a reduction of the mouse myeloid compartment, and that frees up a niche for the human myeloid cells to develop when boosted. And I'll show you how this, what the consequence of that mutation on the development of the immune system. First, let's try to summarize what makes this model different from the existing models. As you know, many models has been used over the last 10 to 15 years, and the main difference is the following. Most of the models available have a SCID mutation. As you know, SCID is a mutation in the core gene PRKDC, which involves DNA repair mechanism. As such, those mice developed spontaneous lymphoma over time, and this has been well documented. In our case, as said previously, we have a RAG2 mutation. So we have no spontaneous tumor which developed in the animal. The mice are pretty much resistant to irradiation when compared to mice having a SCID mutation. And as such, you can use any kind of drugs, including drugs which include DNA damage in these animals. There is no weakness of IgG appearance with age in those mice. And finally, the complement cascade is functional, and the mouse myeloid system is immature, which will reduce the rejection of human tissues when implanted in those mice. How do we use this model for reconstitution? So the strategy that we're using is mainly based on CD34 positive cell reconstitution. We also use PBMC reconstitution, but as you know, when you use PBMCs, your window of action is rather limited, and your immune response will have a very strong bias toward T cell, which include some kind of limitation in the outcome of the analysis that you're going to perform. So we basically humanized newborn animal. We inject CD34 positive cells intrahepatically, and those mice will develop a human immune system if the process went well. 3 months later, we analyze the immune system in these animals. Since we have a knockout of FRIC2, we could boost the human myeloid compartment on demand by injecting recombinant FRIC3 ligand. That FRIC3 ligand would then activate the human myeloid cells, and those human myeloid cells will proliferate. And I'll show you again some data around this in the coming slide. So first, let's focus on the myeloid compartment prior to any FRIC3 ligand, so prior to induction of the myeloid compartment. This is on average what we have. This is basically the percentage of human CD45 positive cells in the animals, and these are the different immune cell population we're seeing in those mice. So we have a majority of B cells, which does not reflect at all the human situation, but that's the case in any HESI model. We have the same in the NSG HESI or in the NOD mice. These are what you should get based on human physiology, and this is what you should get based on mouse physiology. Here we are showing the percentage of human cells, and here you have the count per microliter of blood. When looking now into the T cell compartment, we do see a majority of CD4 T cells. Then comes the CD8 T cells, and these are the double positive and double negative. What happened in the spleen or in the bone marrow? When focusing on T cells in those tissues, we do detect a majority of CD4 T cells. Then come the CD8 T cells and the TRex, and this is the same in both spleen and bone marrow. And we also detect TCR gamma delta T cells in those mice. How long does the immune system last? Here is a paper that was published in 2019, if I remember correctly. And as you can see here, we have been able to detect presence of human CD4 and CD8 T cells up to 51 to 55 weeks post reimplantation. So basically, the model stays stable over time. We do see some fluctuation in the T cell numbers, as you can see in those graphs. However, the human immune system remains present in the animal, and the animal do not develop any obvious sign of GVHD, despite the engraftment, which lasts for a month, more than a year, for a year here. What about now the induction of the myeloid compartment? This is basically how we induce the myeloid compartment. So as said, we inject FLT3 ligand and we investigate the myeloid compartment. So injection of FLT3 ligand is provided over a week. And when we breed the mice right after the injection, this is what we observed. In the FLT3 ligand treated mice, we observe an increase of both myeloid cells and lymphoid cells. When focusing now into the myeloid compartment, this is what we observe. These are the FLT3 ligand treated mice. These are the non-FLT3 ligand treated mice. Here we have mice which are either 19 weeks old in black or 23 weeks old in red. And as you can see, when looking into myeloid cells in general, CDCs and PDCs, we detect an increase of those cell population in both blood and spleen. Similar observation has been also reported and published by Lopez-Lastro and colleagues in 2017. When they look into spleen and bone marrow, they look into total numbers of cells in and percentage of cells. And again, here they were able to see that FLT3 ligand treatment enhances PDCs, CDCs, and monocyte in all tissue tested. What about the other myeloid cells? Here we follow the same protocol and we investigated eosinophils, monocyte, and neutrophils. And again, the myeloid compartment in general is boosted in both blood and bone marrow. However, despite this boosting, the overall number of neutrophils remain quite low in the animals. We decided to look for other targets which are expressed on myeloid cells and which have interest in immuno-oncology. As you know, there is an increasing interest for certain myeloid-specific targets, such as LAIR1, which are known to be associated to metastatic growth in melanoma models. So we wanted to see whether myeloid cells that developed in those mice do also express this myeloid target, and the answer is yes, as you can see here. When we check for LAIR1 expression on T cell, B cell, NK, monocyte, and DCs, we were able to detect it. And we find that LAIR1 was expressed at higher level on affected T cells when compared to resting T cells. Finally, we also detected the target on NK cells. What is important is that the same study has been conducted as well on other targets which are myeloid specific and which are also inhibitory receptors, such as LAIR1, and those targets which are human specific, such as ILT2 and ILT4, was also detected, but I'm not going to show those data today here. Does the FLT3 ligand treatment increases the NK cell number? Answer is yes. We again here run a study where we look for NK cell number in spleen, lung, and the liver. And by looking both at numbers of cells and percentage, we did see an increase upon treatment with FLT3 ligand. Those NK cells, when analyzed for different NK cell subset revealed that we are getting an overall increase of the NK cell population without changing the relative percentage of the different NK cell subsets. Finally, when those NK cells are recovered from the animal and stimulated ex vivo with a cocktail of cytokines such as 12, 15, and 18, we detect a higher production of interferon gamma in the FLT3 ligand, in NK cells recovered from FLT3 ligand treated mice versus NK cells coming from the control group, which was treated with PBS. So the myeloid compartment develops, NK cells develop, and they have a great ability to produce interferon gamma. How long does a myeloid compartment last in these mice? So here we basically did a study which worked as follows. Mice were treated with FLT3 ligand, good, and then nothing happened for 2 weeks, and then we retreated them. And basically what you can see was the following. You can recall, so the first treatment resulted into an increase of myeloid cells, monocyte, PDCs, and CDCs, and you can recall this boosting 2 weeks later, and you get a similar increase. So basically, you decide when you want to boost the myeloid compartment. Similar study could be conducted when mice are reconstituted as young adults. Here is a study conducted jointly with Roche. They took young adult mice, reconstituted those, and looked for the immune cells in those mice. And they were getting similar data to what we obtained. And when looking into the myeloid compartment, they were also able to detect an increase of monocyte, PDCs, and CDCs in those mice when compared to the control animal, which were not treated with FLT3 ligand. So the boosting of the myeloid compartment is not dependent on the reconstitution procedure by itself. It's really model dependent. Could we use this model to assess immune-related adverse effects? I'm going to share with you 2 studies that were conducted by 2 different groups. First, we wanted to know if this model will be relevant to address to see whether compound could trigger CRS. Here is a study published by AstraZeneca last year. In this study, they showed that when using PBMC reconstituted mice, you could trigger a CRS response upon inoculation of OKT3. And that works well. You do see interferon gamma and TNF. And you do see several cytokines produced in these animals. However, when you use PBMC reconstituted mice, you're mainly tackling the T cell compartment. What happened when you use mice reconstituted with CD34 positive cells? In the same study, they used NSG reconstituted with CD34 positive cells. And as you can see, they were not detecting here TNF alpha or interferon gamma compared to the PBMC mice. One of the questions which were underlying those results was whether or not this was due to the fact that the myeloid compartment was missing, and as such, the immune response that was observed upon OKT3 injection was very lymphoid cells dependent. We decided to repeat the same study with CronBio. We reconstituted animals and we injected those mice with OKT3. Mice were bled at 2, 6 hours, and 24 hours post OKT3 injection, and we checked for cytokine production. When the mice are not treated with FLT3 ligand, we detect TNF alpha and interferon gamma, whether this was not detectable in the CD34 reconstituted NSG mice. When mice are treated now with FLT3 ligand, we do detect those cytokines, but at a much greater extent. And this detection really follow the kinetic of cytokine, which is known to appear in the context of CRS. We also detect additional cytokine, which are mentioned here, such as 12, 1 beta, and 18. We treated those mice with tocilizumab, and we observed that when blocking the IL-6 receptor, that mitigates the cytokine response. So, model could be used to assess CRS. The CRS that we're seeing here comes together with a loss in body temperature and body weights, and both parameters, temperature, body weight, but also cytokine release, could be mitigated when using an anti-IL-6 receptor. What we haven't investigated yet were whether or not we are getting some neurotoxic effect and tissue damage in those animals. And that's something that we would love to do. And this is something we're going to do indeed in collaboration with partners. So that's something we're going to discuss at the end. What other immune-related adverse effect could we investigate in the model? In this paper published by James DeSanto and colleagues from the Pasteur Institute, they basically use this model to assess the mechanism which underlie IL-2 high-dose therapy side effect. As you know, in certain type of cancer, IL-2 treatment is used, and high dose, which often result in a higher success, are often associated with certain side effects such as vascular leak syndromes, whereas this is not seen in lower dose of IL-2 treatment. Question was, what are the mechanisms which are responsible of this toxicity? James DeSanto and colleagues used the BRGSF HESI mice, and they treated the mice with different doses of IL-2. And as you can see, when mice receive high dose of IL-2, that results in a loss of body weight of the animals. And they also observed in the same animal a massive expansion of T cell zone within the liver and the spleen, and they observed some pulmonary edema, which indeed mimic the VLS syndrome, which is observed in patients. They look at the cytokines which are produced upon IL-2 injection in the animal, and basically they are having a profile of cytokine storm, which is dose dependent, which respond in dose dependent manner, and which is observed basically both by looking into cytokine but also into chemokines. Finally, they decided to investigate the cells which could be responsible for toxicity. As you can see here, when they inject IL-2, they have an expansion of the absolute number of CD45 positive cells and T cell numbers. And they observed in these mice that upon depletion of CD4 and or CD8 T cells, that results in a inhibition of the body weight loss, which is observed in the IL-2 treated mice. Question was whether this could be due to TRex. The observation was the following: when mice are treated with low dose of IL-2 therapy, that results in an increase of TRex in these animals. When mice receive high dose of IL-2, they are no longer seeing this increase of Treg, but they do see indeed a decrease of Treg. And that results in an increase of cytokines such as 6 and 12, which are known to inhibit Treg suppressive function. The idea beside this was whether this is the reason why they were seeing this toxicity. They decided here to treat the animal with a drug called camphorol. Camphorol is known to preserve Treg suppressive function. And as you can see, camphorol treatment resulted in a diminution of the body weight loss and in an increase of the survival of the high dose IL-2 treated mice. So these animals have Tregs, the Treg are functional, and the Treg activity is tunable depending on the treatment which is provided. What about the anti-tumor response in this model? First question we address is whether or not the myeloid cells could be recruited to the TME. So we did the following study. We humanized some animal. We randomized the mice based on their percentage of different human immune cell subsets, and we injected to those mice A549 tumor cells. Mice were then monitored for tumor growth. As you can see here, FLT3 eigen has no impact on the growth of the tumor. What about the cell recruitment in the TME? Few satellite mice were sacrificed, and we looked into the cells in the TME, and this is what we observed. When looking into cell composition in the TME, we observed, as you can see here, a decrease of NK cells and an increase of myeloid cells and T cells. And when focusing on the T cells, we were able to show that those T cells were mainly CD8 positive T cells. So we not only having myeloid cells produced, but they get recruited at the site of injury, which is here in where the tumors grow. Could we use a model in order to assess combination therapy? In this paper, in this couple of papers, they have used indeed the model as a decision tool helper for combination therapy. I'm just gonna detail a little bit this first paper from this group. In this paper published by Capasso and colleagues, they humanized the animal with MDA-NB231 tumor cells, and they decided to assess the ability of the model to respond to anti-PD-1 treatment. So they treated the mice with nivolumab, and what they observed is that the HES mice do respond to nivolumab, whereas the non-HES mice have no response at all, which is what you would expect. So the immune system is responsive to nivolumab. Then they decided to investigate the immune cells recruited. Here in nivolumab-treated mice, we observe an increase of the frequency and numbers of T cell in the PD-1-treated mice, whereas nothing similar is seen at day 11 post-treatment. That increase comes together with an increase in CD8 T cells in the TILs at day 21. And when looking into the percentage and numbers of interferon gamma and granzyme B secreting cells, they also observe at day 1 an increase in interferon gamma producing cells, so effector cells and polarized cells in the TILs of the mice, whereas nothing was seen at the periphery. The model was then tested here in order to see what happened in terms of TRex response. So in terms of TRex, there is no difference over time in control mice, whereas TRex do not show, sorry, in terms of TRex, we do show an increase in the control mice, whereas in the PD-1 treated mice, we have no increase at all of the Treg numbers. Finally, in those mice, we have a decrease of myeloid cell frequency in TILs, and the frequency of HLA-DR myeloid cells did also decrease, and those cells are thought to be potential MDSCs, although this has not been clearly shown in the paper. Model was used to assess a combination therapy, and basically what they were able to show is that the combination of PD-1 and HDAC inhibitor resulted in a greater tumor growth inhibition than PD-1 alone, and the TILs secrete interferon gamma, and they have more myeloid cells when compared to cells at the periphery. Finally, TILs from the dual therapy treated mice show more activated T cells and a decreased Treg numbers versus monotherapy. So as such, they decided to use this model as a tool to assess different types of combination therapy, and that's one of the advantages compared to the knock-in model that I was talking about at the beginning. Model could also be used for cell depletion studies, and that's going to be the last study I'm going to share with you. This group was developing an anti-PDCs. This antibody targets CD303, which is also known as KEC4C, and that target is PDC specific. And as you know, PDC have been shown to contribute to be, let's say, a poor prognosis for a myeloma progression. So they developed an antibody which was tested first in vitro in HEK cells, and they were able to show that this antibody works throughout ADCC, ADCP, and to a lesser extent, CDC. So this has been well characterized in vitro, in cells, and then they decided to test the antibody in vivo. Mice were injected with this depleting antibody, and they looked for the depletion of the PDCs at different timing. When they looked into the blood, they were able to see a depletion of the PDC starting from day 1 up to day 3, and then the numbers of PDC starting to go up again. Whereas when looking into the spleen, that depletion starts slightly later at day 3 and is also detectable at day 7, and nothing comparable is observed in the mouse. We also have data with another group from another company, which is called eigen, in a joint study we did, where they tried also to deplete cells using different mechanisms, and they were able to show that these animals are able to be, could be used for therapeutics, which work through depleting strategies. So in conclusion, we know that the model here expresses a functional human lymphoid and myeloid compartment. The complement cascade is functional, although I did not really show data on this today. We also have data showing that model could be used for vaccination studies, and it could be used to assess the efficacy of different types of therapeutics, which could be either bispecific or depleting agent. We have also performed some study using compound which targets the toll-like receptor and their partners, and model could also be used as such. Finally, we know that model could be used to assess immunerated adverse effects, as shared with you today. How this model stays stable over time? I just want to share with you here a study that was done jointly with Janssen. They used young adults which were reconstituted, and they compared the body weight of the mice to the NOG-EX cell, which is another model which has a myeloid compartment. And as you can see, whereas here we do see a body weight loss, we don't see this. Why we're not getting this body weight loss, whereas the other model do? It's very simple. In the NOG-EX cell or in the SGM3 model, the myeloid compartment is supported by a massive overexpression of cytokine, and that overexpression could not be regulated because they are using ubiquitous promoter, synthetic promoter, and we all know that cytokine will regulate each other throughout negative and positive feedback, and this is what is lacking. Here, since we're going through a boost of P3 ligand that produce the boost necessary to expand the myeloid compartment, and then the antigen maintain the myeloid compartment over time. Models are reconstituted at genOway, so basically we are based in France, this is where we do this work, but then mice could be shipped abroad, and I'm just going to share with you two sets of data. Mice were constituted here, sent to IGM Bioscience, so in Redwood City, California. And as you can see, mice were QC'd here and QC'd upon arrival. No differences. When we ask them to induce the myeloid compartment, just to make sure that the inducibility remains functional, they were able to induce a myeloid compartment here, which we haven't done here. Finally, we did another study, as I said, with Janssen, so East Coast, and mice were able to be shipped there, and we had no modification of the body weight loss over time, so the model could be shipped wherever it is. What is next to come with this model? So we would love to go a little bit further in the ability to use this model in order to assess immunerated adverse effects. For that, we would love to go further in our study on CRS and ADA and go and see whether we do see changes in tissues upon induction of this type of mechanisms. See whether model could be used as and standardized using drugs which are already in the clinic, drug for which we already know their tox profile, and finally investigate the donor variability in this context. Check whether the myeloid compartment immune response works as expected with drugs which are known to target this compartment, and finally do some precision oncology in this model, which is something we already started. We are also interested to investigate the use of the model in standard of care, and for all of this, we are open to discussion with the member of this workshop and see what could be done jointly. Finally, which are the improvements to come? We are generating this model. In fact, we already have the model with a W41 mutation in c-KIT, which should enable to overcome the irradiation step. And we are at the moment making this model onto a HESI FcRn double humanized background. We already generated a model which is a knock-in model in which both FcRn and HESI are regulated physiologically. Model has been used successfully for different type of study, including biologics which have either an extended half-life throughout HESI binding or throughout FcRn binding. This has been published. We are using the same strategy to humanize HSA and FcRn onto the BRGSF background, but that's something that will probably be available in a year and a half from now on. So I just wanted to conclude by saying thank you to the team here at genOway who worked on this model. And more precisely, Florence for the work done around this, and Thiam for help in putting this slide together and the work around this model and the knock-in models, and all our partners from TronBio, Antineo, Janssen, IGM, and Roche who have worked with us on the validation of this model. Thank you.